Exploring Life Chapter 12. Evolution of Genome. Part 1. In this course, we often talk about evolutionary changes, so now it's time to look at the details that drive these processes. Genes are specific sequences of DNA that contain the instructions for making proteins. Every gene serves as a blueprint for a particular protein, and the number and size of genes can vary greatly between different organisms. Altogether, an organism's genetic material, including its genes and all its other regulatory DNA, is called the genome. Gene expression regulation controls the timing, location, and amount of protein production in cells. Unlike multicellular organisms, which can exhibit complex behaviors and decision-making processes, cells do not act randomly or with conscious intent. Instead, cellular responses are governed by regulated changes in gene expression, which are determined by encoded regulatory elements and molecular networks within the genome. Passing on genetic information is a fundamental property of most cells. Exceptions exist. For example, some specialized cells in multicellular organisms rarely divide or do not divide at all, and germ cells follow meiosis to produce gametes. But faithful transmission of genetic information is a central feature of cellular life. Together, these tightly regulated processes, gene expression and faithful genetic transmission, allow cells to grow, adapt, and contribute to the incredible diversity of life. Despite sharing the fundamental similarities just discussed, prokaryotes and eukaryotes have very different approaches to genome organization. By understanding these contrasts, we can gain insight into how they evolved, what underlies the remarkable success of prokaryotes, and how greater genome complexity supports the increased cellular and functional diversity of eukaryotic life. The first striking difference is genome size. Prokaryotic genomes are often thousands of times smaller than those of eukaryotes. They are compact, containing hundreds to a few thousand genes across around 500,000 to a few million base pairs. For example, the bacterium Mycoplasma genitalium has a genome of just 580,000 base pairs and about 525 genes. In contrast, eukaryotic genomes contain multiple thousands of genes and range from millions to billions of base pairs. The human genome contains around 25,000 genes distributed across 3.2 billion base pairs. And some eukaryotic genomes are truly immense. For instance, the flowering plant Paris japonica has a genome of about 670 billion base pairs, most of which is non-coding DNA. This enormous variation in genome size between prokaryotes and eukaryotes reflects the greater biological complexity of eukaryotic life. As we learned earlier, prokaryotic genomes are typically composed of a single, circular DNA molecule. This compact structure matches their relatively small genome sizes and requires minimal DNA packaging. In contrast, eukaryotic genomes are divided into multiple, linear chromosomes and rely on highly organized DNA compacting mechanisms. This transition from a circular to a linear genome is considered a major evolutionary advancement. Some researchers hypothesize that this shift may have originated from the accidental breakage of circular DNA molecules. Over time, these changes were selected for and refined through evolutionary processes. The resulting linear chromosomes, tightly packaged with proteins, provide greater structural stability during cell division and allow for the accurate and precise inheritance of much larger, more complex genomes. In prokaryotes, the genome is located directly in the cytoplasm, without a surrounding nuclear envelope. Prokaryotic genomes are relatively simple, with very little non-coding DNA, so most of the DNA has a direct functional role. Many prokaryotic genes are also organized into operons, clusters of related genes transcribed together, allowing the cell to efficiently coordinate their expression. This streamlined genome organization is one of the key factors that enables the rapid growth and remarkable success of prokaryotic life. In eukaryotic cells, the genome is enclosed within the nucleus, and most genes are regulated individually rather than in operons. Each gene is split into protein-coding segments called exons and non-coding intervening sequences called introns. Introns often contain important regulatory elements that help control gene expression. They also enable alternative splicing, a process that allows a single gene to produce multiple protein variants by combining different sets of exons or retaining specific introns. This complexity supports the increased cellular and functional diversity that characterizes eukaryotic cells. The schematic summarizes the key differences between eukaryotic and prokaryotic genome organization that we've discussed. The differences are closely tied to genome size and reflect the varying complexity of these organisms as well as their evolutionary history. From this point forward, we'll focus exclusively on eukaryotic genomes, like the human genome shown here. In the upper section, we see the cell's nucleus with chromatin in its relaxed, extended form, which allows for gene expression. In the lower section, that chromatin is condensed into the familiar chromosomes that appear during cell division, ensuring the accurate preservation of DNA. Next, we'll explore how eukaryotic cells maintain a delicate balance between protecting their DNA and keeping it accessible when needed. Eukaryotic chromosomes are linear, thread-like structures made up of DNA and associated proteins. Some of these proteins help with DNA packaging. The first level of this packaging involves small protein cores called histones. DNA wraps around these histones to form nucleosomes, like tiny spools around which the DNA is wound. The nucleosomes then coil and fold into chromatin fibers, compacting the DNA while keeping it flexible enough to be accessed when needed. This more relaxed packing is typical when a cell is actively growing and functioning. However, as the cell prepares to divide, its DNA becomes much more tightly packed to ensure that the genetic material can be accurately and efficiently distributed. This illustration shows the hierarchical packaging of DNA inside the cell. Starting at the bottom, you can see the DNA double helix. First, the DNA wraps around histone proteins to form nucleosomes, the fundamental units of chromatin. These nucleosomes then coil into a 30-nanometer fiber, creating a more compact chromatin structure. Next, protein scaffolds fold this fiber into looped domains, packing the DNA even more tightly. Finally, as the cell prepares to divide, the chromatin is condensed all the way into fully packaged metaphase chromosomes, the most compact and visible form of DNA in the cell, as shown at the top. The metaphase chromosomes appear as fuzzy, rod-shaped structures. At this stage, the DNA is so densely packed that it's virtually inaccessible. Let's use an analogy to better understand DNA packaging and its implications for gene accessibility. Think about your kitchen when you're cooking. Everything is neatly arranged and within reach, so you can quickly grab a pot or spoon whenever you need one. But when it's time to move houses, you have to pack everything into boxes. It's safe, compact, and easy to transport, but much less convenient if you suddenly need to find something. DNA works the same way. When a cell is active, its DNA is loosely organized and easily accessible so the cell can use its genes whenever they're needed. But when it's time for cell division, the DNA is tightly bundled into chromosomes, securely packed for safe transport and accurate distribution, even though it can't be easily accessed until it's unpacked again. After DNA replication, each chromosome consists of two identical sister chromatids joined at the centromere. Each chromatid is a complete copy of the chromosome. The sister chromatids separate and become the individual chromosomes during division. Before the next division, the DNA will be replicated again, and the process will repeat. This illustration compares a chromosome at different points in the cell cycle. On the left, the chromosome is shown before division, consisting of two identical sister chromatids joined at the centromere. On the right, the chromosome is shown after division. Once the sister chromatids separate during division, each is considered an individual chromosome and no longer consists of chromatids. It will be replicated again before the next cell cycle. Now let's look at how the cell performs one of its most important tasks, protein synthesis. The central dogma of genetics outlines this process, which occurs in two main stages, transcription and translation. Transcription, where DNA is copied into messenger RNA, takes place in the nucleus. Then, translation, where the mRNA is decoded into a protein, happens in the cytoplasm. This figure illustrates the flow of genetic information. At the top of the figure, DNA is transcribed into messenger RNA, or mRNA. The mRNA then travels to the cytoplasm, shown in the middle, where ribosomes and transfer RNA work together during translation. At the bottom of the figure, you can see the resulting amino acid chain, which will fold into a functional protein. And to help make this clearer, let's use an analogy. Think of the DNA in the nucleus as an irreplaceable instruction manual. You wouldn't take the entire book into a workshop, where it could easily be damaged. Instead, you'd make a copy of just the pages you need. That's like making mRNA. Then you take the copy into the workshop, the cytoplasm, where you follow its instructions and use your tools, the ribosomes and tRNA, to build the final product, the protein. It makes perfect sense now, right? Transcription begins with initiation, when RNA polymerase binds to the promoter region of the DNA. DNA consists of two complementary strands, and different genes use different strands as their template. The promoter sequence of each gene determines which DNA strand will serve as the template. Once bound, elongation follows. RNA polymerase unwinds the DNA and adds complementary RNA nucleotides to synthesize the RNA strand. Eventually, RNA polymerase reaches a terminator sequence and releases the RNA transcript. This process produces a pre-mRNA molecule containing both exons, the coding regions, and introns, the non-coding segments. Finally, post-transcriptional modifications occur, introns are removed, and exons are spliced together to produce a mature mRNA ready for translation. Using our earlier analogy, we've just described the copying process and how the instructions are edited to be more concise and easier to follow. This image shows transcription in action. The two strands of DNA run in opposite directions, one going 5' to 3', the other going 3' to 5', kind of like two-way traffic on a street. RNA polymerase, shown in green, moves along the DNA and builds a pre-mRNA strand, shown in red, by copying the genetic instructions encoded in the DNA. Translation occurs on ribosomes in the cytoplasm, where the mRNA sequence is read and turned into a protein. Transfer RNA, tRNA, brings specific amino acids to the ribosome, matching them to the mRNA codons, sequences of 3 nucleotides. With 4 nucleotides available, 64 codons are possible based on all potential combinations. The process begins at a start codon AUG and ends when a stop codon UAA, UAG, or UGA is reached. As each codon is read, the ribosome links the corresponding amino acids together, forming a growing polypeptide chain that will eventually fold into a functional protein. And again, going back to our analogy, we've just described how the instructions are followed and the final product is assembled in the workshop. This illustration shows the process of translation occurring in the cytoplasm on ribosomes. The ribosome moves along the mRNA strand, marked in red, reading its nucleotide sequence. Transfer RNAs, tRNAs, shown in green, match each codon on the mRNA with the correct amino acid. These amino acids link together to form a growing protein chain, as shown on the top left. This process translates the genetic information carried by the mRNA into a protein. The genetic code is highly efficient and universal. Even though there are 64 different codons, they specify just 20 amino acids, so most amino acids are encoded by more than one codon. Despite this redundancy, each codon unambiguously codes for a single amino acid. Remarkably, the genetic code is virtually the same in all living organisms, underscoring our shared evolutionary history. You can think of the genetic code like a universal language, with codons as words that carry the same meaning across nearly all species, from prokaryotes to humans, fungi to plants. Its consistency highlights the deep connections among all forms of life and supports the idea that we share a common evolutionary origin. This is the translation table that matches codons to their amino acids. To use it, look for the first nucleotide of your codon on the left, the second across the top, and the third on the right. Where these 3 meet is the amino acid that codon encodes. You can think of the genetic code as a universal language that all living organisms understand. It's made up of 64 3-letter words, called codons, and these words specify 20 different amino acids, the building blocks of proteins. Just like in any language, there's some redundancy, so multiple words can mean the same thing, similar to how apple, fruit, and food all relate to something edible. But there's also precision. Each codon always corresponds to one specific amino acid, so there's never confusion. Apple will never mean table. You can also think of the genetic code like a sentence. The start codon, AUG, is like a capital letter that tells the cell where to begin reading. After that, the codons flow together without spaces, like a string of words. Finally, the stop codons, UAA, UAG, and UG, are like punctuation marks at the end of a sentence. You can picture them as a period, a question mark, and an exclamation point, telling the cell that the message is finished. In this activity, you'll practice thinking in the language of the genetic code. On the top left, you'll see a quick summary of the concept. Every sentence in this language begins with a start codon. Like a capitalized word, we use the word the. Each codon is a 3-letter word, and these come one after the other, without spaces. Finally, a stop codon acts like punctuation. We use the word end. It tells the ribosome to stop translating. On the top right, there are 2 example sentences. Both begin with the, include a string of 3-letter words, and end with end, like the cat ate bat end. The dog run far end. Now it's your turn, just follow the instructions on the bottom left to craft your own message. Have fun creating silly sentences, and remember that everyone and everything understands what you're saying in this genetic code language. In the second part of this lecture, we'll examine how DNA is replicated and explore the kinds of mutations that can arise during this process. Now it's time to check your understanding. This self-evaluation will help you review the key concepts we've covered and assess your progress.